And this lecture is kind of an introduction on where the thinking behind this module comes from. Why have we given you a project to do, some of your own science to kind of take on and take the lead on? Okay, so two things we're going to do is just deal with some basic issues of kind of philosophy of science. I don't think there's much exposure to philosophy of science. And then the other thing is some ways of thinking about how to explore the topic. So, how does science work? And I mean, when we're thinking of science, how does it actually, what is the model that we use to think about it? And this is the kind of traditional ways, like we have time on our x-axis, we have knowledge on our y-axis, and it's just an ever-progressing increase. How many of you kind of think of science in this way? I mean, we're always increasing the bounds of knowledge. Yeah, it kind of makes sense. Yes. So maybe, yeah, go through a world war and then it'd be a spike, and then afterwards there'd be a dip or something like that. Sorry? You know that you know less by knowing more. It's pretty deep and insightful, right? Yes, yes.
So when completely new ideas arise, then some of the knowledge has to start over again.
So we're starting to get to this core of, is there always an increase in knowledge? So the other way to think of this, in fact, let me ask you a question. Where is that bit of knowledge? Where would that fit in here in terms of the world is flat? The world is flat. Where would that fit in here? So at some point, the part of the knowledge, at some point, it was given that the world is flat. That was part of the knowledge base. Okay? But here we say that knowledge is increasing. But there's this point that what we know now might be wrong. So we might think we know a lot about a lot of things. Okay, and so we're marching along here. But actually, most of what we know might be wrong. And so how do we then deal with that? There's another conceptual model of the way that science works. Is that there's a community of people, of researchers, whoever it is, they're interested in a topic, and they're advancing that topic. This could be mathematicians, this could be biologists, this could be pollination biologists, it could be very, very specific. It could be people who are interested in restoration or some weird derivative of math. There is this community of scientists who have an understanding about a topic. And that community, their understanding of the topic is built on the previous community's understanding, or that same community but their earlier understanding. Somehow they've shifted from here to here. They've shifted their understanding of the topic. And they might be going on, what we know now might be completely wrong, or we need to expand and shift into a new sphere of knowledge. So this idea that we're just always increasing knowledge is not, try not to think of science in that way. The way that we're trying to get at this is to move this community of knowledge over to that spot where actually it's true. But it's very difficult to figure out where that spot of truth is in the landscape. So, as a researcher, what is your job? What do you think your job is?
Push the boundaries.
To make these boundaries bigger. Okay, push the boundaries. Yeah. Wherever, or shift the boundaries. Shift the thinking. And it's not necessarily, you don't necessarily have to shift shift the boundaries. It could be to reinforce what we know already. So we know something already, and we do a completely new test to show that what we know now is still correct. So that's also very useful, to do additional tests to say that the knowledge that we know now, we look at it in a completely new light and there are bits of another technique, different lines of evidence, and what we know now is still relevant. Or hang on, this test was designed to prove that what we know now is correct, but actually it shows that what we know now isn't correct. So your aim is to engage with this community of researchers and either shift their thinking or increase their thinking. But all of your research needs to be conducted within a community. You've got to change how they think. You can't, if you just change the way that you think, the way that you understand the world, I mean, that might be your primary objective. You want to understand the world better, that's fine. It's great. But if you come up with a whole new theory and you don't convince your fellow researchers about this theory, you don't tell them about this theory, have you done anything to the bound of human knowledge, the bound of science? No. So it's not just about you coming up with new ideas and testing the ideas. You've got to be able to convince that community. So I just always want you to keep these kind of two images in your mind. Because for you to convince that community, to go and add a little bit of knowledge is easy. I think I used the example last time of to generate knowledge is easy. We could count the number of people in this room, and at this given point, at this time, that's new knowledge, yeah? But it's not valuable. You go and take that to whoever your community is of researchers, and they're going to say, so what? We don't care. So you've got to find a bit of knowledge that they care about, that either like, oh, that's interesting because that supports our hypotheses, or that's interesting because that completely disproves our hypotheses. Now we're going to look at something new. So the increase of knowledge and the way that it can only happen when you're thinking about the community that you're going to be with. So the other problem with this is that this assumes what we know right now is right. Whereas if you think in this way, then it assumes that, for those that at the back there, it's probably a bit small font, so it assumes that what we know is bits that are right and bits that are wrong. We need to keep the bits that are right and replace the bits that are wrong. So we've got to reinforce what we know is right, figure that out, and we've got to figure out what is right and what is wrong. Get rid of the bits that are wrong. So there's always this, these two terms bandied about in terms of research. It's got to be new and original. You've got to be doing new and original research. What do you think about that in terms of these two models that I've given you? No? It's got to be new and original. No? How many of you think that all research has to be new and original? Hands up. Okay. If I ask the inverse of that question, am I still going to get no hands? So, just by going back and repeating someone's study, it's not new and original. But then you get the same result. It's not new and original. Is it valuable? Hells yeah. Okay? So please, when you're looking at your projects, don't feel that you're under obligation to come up with something that's new and original and increasing the knowledge in science and standing on that island of knowledge and building another sandbar into the ocean of ignorance. Those kind of metaphors don't help you. What you want to know is figure out, what do people know about my topic? Is there some way that I can confirm what we know is right? And maybe, which would be pretty cool, what we think we know is wrong. That would be pretty neat. But either way, it's valuable. So, focus on value. And all of you at this level can do valuable research. That's a crucial point for you to understand. Well done, you braved the maddening
Didn't
hordes.
understand what position to go.
Congratulations. Thank you. Okay. Some of you will come across this point with your project. I can't find my community. You know, my project is so specialized that there is no community. You're the only sole person that has any interest in this particular topic. I would argue then that you really need to think about whether your research is valuable. If you're the only person that's really, really interested in the number of people that are in this room right now, the urgent need to record that, you need to think, well, is the work that I'm doing valuable? Because there must be a community of people out there, researchers that have an interest in this. So, I've looked, and there's no. Try and not. So this often happens if your topic is on like a given species. The seed production of Metalasia muricata in a summer in a drought. So what you do is you do your search for Metalasia muricata, seed production and drought, and you find that no one else has looked at those three things, and therefore you're the only person that's potentially that has ever thought about this and is interested. No. Metalasia muricata is part of dune flambours. It was part of the flambours, so immediately you fit into the flambours community. You're interested in seed production. And so now you're into the broader community of like seed production of wind dispersed species versus bird dispersed species. So you can then under drought, where there's a lot of people who are interested in environmental change and what effect the droughts will have on it. So you've got to then figure out, is there anyone who kind of intersects with those larger flambours, environmental change, maybe some kind of aspect of dispersal. I can guarantee you will find those people that are interested in those different topics. But it's very, very unlikely that there's going to be someone who's done exactly what you are proposing to do. So the way that you've got to think about this community is you've got this bit of information, or you've got this idea that you want to do, which community is this going to affect? And you've got to go and who would be interested in this? And then you've got to go and interact, you've got to find them and then interact with them. So how do we identify that community? How do we, that's quite a, I'm saying that it definitely exists out there, and you've got to, and so reading the literature is your first port of call, definitely. Okay. Who else? You've got to identify the general fields that your project is in. Look at the literature. May I suggest that while you're going through this phase of trying to find the community that what you want to do is interested in, skim read. Please do not waste time reading a paper from beginning to end. You can skim read 30 papers in an hour versus one paper in 3 hours. You read it intensely. What you're trying to do is find topics, people who have written on topics that are similar to yours. And when you skim reading, you kind of have two piles. One that's yes and no, and then you just carry on. Then you go, once you have your yes pile, or your folder on your hard drive, or reference, however you're going to be doing this, you go through that again and you look for common trends as to what who's more closely aligned, which ones are very close aligned, which ones are very far away. Okay. Any other suggestions in terms of finding this community that your project and your research needs to interact with? Pardon? Other students? Well, you can ask ask other students, yeah? Ask your colleagues. Your supervisor should be able to tell you who your community is. How do you go and ask your supervisor in a nice way? Please tell me who my community is. What the hell are you talking about? Sneaky way of finding out from at least getting a head start on figuring out your community. Go and ask them for the one paper they think is most relevant to the study that you need to do. And what do? You look at those authors. You go and look and see what those authors have published. You go and look at their references. You see who they've cited. If you find a good paper, if you go find a paper, if you find in your skim reading, you find a good paper that like really, this talks to your project. You look at the authors. You see what else they've published. And you go and look at their reference list. Rather than, because that immediately takes you into this sphere. Yeah? Whereas if you're in Google and you've typed in your 5 search terms, you've now got 300,000 hits that you've got to skim across the top. And some of them will be completely unrelated. Levels of relatedness to a project will vary wildly. As soon as you find a good one, use that to delve into this community. Anything else? So you can ask your supervisor. Okay, I don't have all. Those are all my suggestions. I'm keen up. I'm waiting for you. Yeah? No? No? There is one other place that you can look. Scientific blogging is becoming a thing. A lot of scientists are asked to blog about their research. So if you find some authors that are within this, you think they're in this community, go and read their blogs. Much, much nicer than reading their papers. And secondly, if they've written a blog that's really interesting and related to your project, and you write a comment or a question or for more information, they'll pop up in their inbox, and I tell you what, they'll put everything else aside to go and respond to your question.
Why?
Because it's coming. It's not their students bothering them. It's not. They don't tell them you're a student. Yeah. Trick number 1: don't ever tell them you're a student. They've already got their own student issues. You're an interested researcher. Okay. You are not researchers. Don't tell them you're a student. Okay. But whenever I get, on the very, very few occasions that anyone ever reads my blog, and I do get I get an email, I'm like, oh, this is pretty cool. And I respond to it immediately, unless it's completely. That's how they interact with scientists that you think can help you think about your project. Okay, don't just rely on your supervisor. Your supervisor can help you, but your supervisor is only one member of this community, and you need to expand your understanding of that community. So I said, okay, so I had blogs and I said I was a supervisor, and I was hoping to get. If anyone comes up with another technique, please let me know. I'm very interested in trying to figure out how to help people understand this community.
Do you know if there are any scientific forums?
They used to be really popular in kind of mid-90s to mid-2000s, and they just seem to have faded away. ResearchGate is possibly now that replacement, but ResearchGate is like, there's a question and then people that kind of tag have interest on that question, and it'll move, but it's not, like forums are very much like memberships and groups and whatnot. Those seem to have fallen away. So that's another place. Go and look at ResearchGate for similar questions that might be asked. Yeah.
You can use social media.
Yeah. Stalk your literary hero.
But there are a lot of magazines, they have like a very broad, like the ad for a National Geographic, they have a very broad of like content, and sometimes something is asked and you might click the link and a link or article and then there you like go.
Okay.
Stalk everything.
So yeah, go and if you've got a topic that's got lots of public appeal, So it's now been written as a National Geographic article. It's been written up as a conversation article. What does that mean? That this is this community writing for the general public, which is just a broader community. It's just that community expanding. They're trying to interact with, and those are really useful for figuring out, for drilling your way down into this community. There might be many layers to this community. There might be the core scientists, and then you've got what their students think, and some kind of periphery, and then you've got the general public. And then this community might be trying to interact with that general public. That's a good way of getting into this. Any other suggestions? Oh, that's quite good. Okay, so we're going to kind of pause there for a moment and shift into another gear. And I just want to discuss the types of research, because as you're going through your project's going to fit into kind of, there are two major categories or approaches to doing your research. And I just want to make sure that you're comfortable with where your project is heading, in what approach it's using, and why. And the reason why is that I say, okay, so this is the descriptive approach, and then there's the hypothetical deductive model, which we've been hitting you guys over the head because that's what scientists do. Generate a hypothesis, go and test the hypothesis and see whether it's true or. But the descriptive approach is also good science. So the descriptive approach could be something as simple as describing a plant species. Do you need to generate an elaborate hypothesis to test to be able to describe a plant species? No. But if we didn't have plant species, we didn't have those descriptions, we wouldn't be able to use them in later hypothesis generation. If you want to describe vegetation types, do you need to generate a hypothesis? No, you go through a process, a purely descriptive process of going, sampling, and running. You run analyses to describe vegetation. It's very complicated analyses, but it's not the hypothetico-deductive approach. So I just want you to know it's completely fine to follow the descriptive approach, because that is the foundation of being able to generate a hypothesis. Once you've described a whole bunch of vegetation, say across South Africa, now you can say, well, the vegetation changes across all these different environmental gradients. My hypothesis is that it's driven by seasonality of rainfall and temperature and something else, and I'm going to design an experiment to test that. So I take fynbos, lift it up, or try and grow fynbos seedlings in the summer zone, and try and grow some acacias from eastern savannas in the Cape Forested region. But that's emerged from the vegetation mapping that's taken place, the descriptive approach. Okay, and some of your studies might just be descriptive. So when you're coming down to like the hypothesis, and you really are battling to do it, please don't try and shoehorn, I'm going to identify this species into a hypothesis-driven framework, because it really just hurts. To read that just hurts. Okay. But then on the other hand, we have the hypothetico-deductive approach, where we've been driving that this is, you've got to have a hypothesis, and it's got to be falsifiable, so you can collect data and see whether it falsifies the hypothesis, or whether the hypothesis is consistent with the data. So that's one way of doing it, and that's usually when you've got one hypothesis and one idea. The other thing that I want to introduce you to is something called multiple competing hypotheses. Because if you can get your project to this point, if you can get your understanding to this point, it's a very valuable way of thinking about the world. It's a very valuable framework of testing generating multiple ideas that could apply to a system, and finding ways to see which idea is correct. So I'm going to give you an example of all of these three. So this is a topic that I've been interested in the last few years. We've just published a paper now for the first time describing what a one plant is. And do you notice this very thin stemmed plant? It's completely leafless. And so these got leaves and flowers at the top. These things are common features in the Cape landscapes. We find them in lowlands, we find them in uplands. We find them in wet areas, we find them in dry areas. They're all over the Cape Coast region. It's a unique architectural trait that you don't find anywhere else. It's really rare to come across these kind of one architectural plants. So we're interested in, we've just kind of kick-started the research on this. So there's they really are very cool and incredibly thin. It's just like a really strange architecture. So this is a Restiolag, and that's an Erica. Most Ericas don't look like that. Here's a Anganon deformer, and this is really weird because it belongs to the family Apiaceae, which is the carrot family. Yeah, so mostly everything's got rootstock underneath and it's got an annual growth form above. And here's this woody thing that every time I looked at it in the famous, I thought it was a pine tree. The first couple of times I saw it until I found it in the book, and it's not a weird looking pine tree. It's an endemic genus to the Cape. This is a Ligula. A little bit more branching, but you can see all the flowers at the top. So what in the world, and this is a year, oh yeah, let's just see if this works. So this is just kind of showing they're all really super bendy. You can flip them over, but we'll just keep on returning to. So this is Lauren Bailey's honours work. She led this project, which we just published now. It should spring right back up. So they're super springy. Wow. And again, all these bare stems. Okay, so descriptive research. Firstly, it's important to generate hypotheses. So the first thing, the descriptive approach. This is actually another species entirely that's growing up. So what we could do is measure the traits of the one plant and versus the other species in the community to say that, look, really, really long branch to branching ratio. Okay, so the number of branches across its length versus everything else that's in there. I mean, this is Leucodendron spignon. That thing's already branched 5 times across a branch, whereas these things have branched 3 or 4 times across that entire length. So, that's entirely descriptive. What we want to say is, like, how does this architecture differ from the community architecture? There's no hypothesis behind it. We just want to show that these things that are visually different, how are they different? So we can measure height, where the leaves are placed, the percentage of their stem across the whole thing, across the degree of branching. There are a whole bunch of traits that we could, just to say that, like, this is an architectural form that is very different from everything else. Purely descriptive. But then, this important thing that can be used to generate hypotheses is, so the hypothetico-deductive approach is that we've got to be able to, remember, you've always got to generate a hypothesis that is falsifiable, that you can prove is wrong, that you can collect data on to prove it's wrong. So, hypothesis. One plant architecture is derived from a shared common ancestor. Zoom with me recently. You should know what a shared common ancestor is, but effectively that's saying that back in time, an ancestral plant grew this one architecture and then diversified, and then that's why we have so many species with one architecture. So our expectation is, and from that hypothesis we can generate expectations, that all one plant species should be closely related. Then we can go test that with the data that we've collected descriptively. So these are the one plants that we know of at the moment. This table, we keep on adding to it on a monthly basis when we find new species. And so the important thing to see here is, so these are the one plant species we've got, these are the genera they belong to, and these are the families they belong to. So they spread across a whole range of families, a whole range of genera which are very species rich. So this is Athanasia pinnata, and it's got 36 species in it. Where's the Erica? That should be impressive. There we go. Erica, 680 species, and there was that one I opened with this Erica fascicularis. Now, of 680 species, we know there's one. We found one plant. So, whatever this is, the reason for this architecture is, it's not from a common ancestor. Nice, simple hypothesis, and we falsify it. Then we got okay. We could also generate another hypothesis to say that all species, maybe we falsified one hypothesis. Maybe all species are found in the same environment. Okay, maybe they're all found in wetlands or in uplands or in lowlands. I've already told you earlier that we find these things everywhere. And so what you could do is actually classify all the environments that you find it in, and you can falsify that hypothesis. And so these things are found across a whole wide range. So we know that it's not common inheritance, and we know that it's not an environmental driver causing convergence. So this is where the multiple competing hypothesis model comes in. And I really encourage you to try and read up on the multiple competing hypotheses framework. It's a very, very powerful way of doing science. It's not always applicable. So again, don't force your research into this if it doesn't work. Okay? But when you can get it to work, it's pretty darn cool. So, we might have a general question. How did this one plant architecture arise? And we could have a number of hypotheses. There's another reason why this multiple competing hypotheses was generated. Before you even look at the data, before you even generate a way of testing the hypotheses, you kind of brainstorm all the potential ways that could generate this plant architecture. Yeah? And then you come up with your list of hypotheses. And then what that list of hypotheses can do is help you generate tests that will then differentiate between those hypotheses. My problem when I went into this study was that I was really convinced it was this. So hypothesis 1, to limit or avoid rodent florivory. And florivory is the eating of the flowers or the seed set. Okay. Fangos nutrient poor. So the leaves are already poor nutritional values, and flowers, the plants invest a lot into their flowers and seeds, so one would imagine that those are little hotspots of nutrition on the landscape that then could be targeted by rodents. Remember, there are not many large mammals, not many large herbivores in fangos, but there are a lot of rodents. The density of rodents is incredibly high. So I was really going for this to limit or avoid rodent florivory. So we set up experiments where we had a one plant that was loose and another one plant that was fixed. We had laboratory mice that were, our expectation was that they would climb to get the peanut butter of the fixed one plant quite readily, but the one that was loose and waving around, they would feel really scared to go up and whatnot. laboratory mice that we thought wouldn't be able to climb these things, were really worried that they wouldn't be able to climb, were up and down these bloody one plants all day long. And in fact, there were a couple that showed that they liked the wavy one. It was going because it was really kind of
Fun.
It was fun and exciting, yeah, exactly. And so I was like, damn, that doesn't fit with our hypothesis. Yeah.
I would actually think if I was a rodent, I would just chew through the stem.
Chew through the stem. So there are examples of that happening in the fangals, where there are restiers that, where many restiers are dioecious, so you get males and females, and the females that have got a big nut. And you find if you're walking through a population of restiers, all the male plants are fine, and all the female plants look like they've been hedge trimmed. Like they've just been, like a brush cutter's been taken through. All the rodents are going on, nibbling along, and the coal falls down, and then they grab the nut and run off with it. These things are really thick and wouldn't get across. Yeah, so it's actually quite a lot of effort to be able to push it down. So that we think it's not that. So then eventually in our paper we limit this and we discuss all of this, all of these things as potential hypotheses. But now I'm returning to this and I'm setting this up in the multiple competing hypothesis framework, where we've got all of these hypotheses to avoid rodent florivory. This one was thrown in by a reviewer to say it's limit or avoid herbivory by now extinct herbivore. And that's because in New Zealand, there's something called divaricating architecture, where these plants kind of shift, they branch multiple times and they're very, very springy, and that's because of the extinct flightless birds. So these big giant birds, but it's very difficult for them to eat off the spring architecture. And so anyway, that's just because of the one example that's in there. To overcome competition for light, why would you grow tall? Why do most plants grow tall? So that they can get light. To increase pollination success, maybe if you're taller, you'll be more visible to the pollinators. This is one that was suggested by a physiologist to increase the cooling, the leaf level cooling on hot windless days. And what that is that if you just move slightly, that boundary layer that builds up around the leaf can break down and then your leaf can cool. But if you're just sitting there and there's no wind, a plant doesn't have a way to cool down. And that boundary layer builds up and then the leaf gets hotter and hotter and then photosynthesis stops. And then another hypothesis is you're so tall so that you can increase your seed distance dispersal. So, to limit or avoid rodent florivory, there's some expectations and we can start spitballing out. There should be lots of rodents. So we should go and check that there is literature that says that in certain types of fangals, there's lots of rodents. But maybe we need to go and check in all the veg types that these one plants have. There is actually a high density of rodents, enough to be able to drive this architecture. Rodents eat a lot of these seeds and flowers, so we can go and conduct tests. We can put out flowers and see how rapidly they're eaten. We can take some of these flowers, drop them to the ground and see and monitor them with camera traps. Rodents would find one plant difficult to climb, or we've shown that they don't find it difficult to climb. But there's another thing that we haven't tested, which is rodents might not want to climb this due to the fear scape. As soon as you're climbing that, you're visible from a long distance away, and any raptors in the neighborhood would come and catch you. So there might be a fear scape component. Still haven't figured out a test for that yet. Well, I was thinking of buying some stuffed rodents, some stuffed mice and putting them on there and seeing if any raptors get fooled or not. But maybe we should, I don't know, something like that. So this hypothesis was suggested by a reviewer. It's really difficult to generate expectations, but at least in this framework, I can say, here's our hypothesis, but what do we do with it? We can't really do anything, but it remains there. It remains in your mind, so that you don't just put it aside. To overcome competition for light, well, there should be high degrees of shading and understory, so you can quickly go and monitor how light changes between up here and down there. You should be able to show their photosynthetic rates decline. A bunch of other things. To increase pollination success, flowers that are high above the ground should have higher rates of receiving more pollination visits. So there should be more seeds set. There should be more seeds set per flower, and also importantly, if it's visibility for increasing pollination success, then there must have animal pollinators, insects or birds or something that can move and see them. Because wind pollination would be quite difficult. I mean, if it's wind pollination, we expect a lot of pollen to be produced. I mean, the immediate thing that you could rule out for wind pollination is that it would be difficult to produce the amount of pollen to be able to get to another plant, which is possible. So we just got to look at that. But if we do find wind pollination, we know that it's probably not to increase pollination success via animal vectors. To increase cooling in hot environments, there is listed expectations, and seed dispersal, they shouldn't have animal dispersals, there should be wind blown or simply drops, so that distance means something. Because if most of these are with animal dispersal vectors, it makes no sense to do so at all. Do you see? So, and some of these hypotheses would have the same predictions. If pollination success increases with height and decline in rodent florifery increases with height, how do you use height to differentiate between these two hypotheses? You can't. So you've got to start looking for something else. What if, as you drop those flowers down, your pollination success increased, but maybe your pollination success should increase and your rodent herbivory should increase. Both of those are correct. But if you drop it down and pollination success doesn't increase, but your rodent herbivory does increase, then you've got support for one and not for the other. I would really encourage you to read more about this multiple competing hypotheses, because it can really help you formulate a framework for understanding your system. And so the way to think about this is to generate alternative hypotheses. You have a system, you have a question. Remember that your hypothesis is always question driven. You have a question. What is this? What is the answer to this question? You've got a bunch of hypotheses. Don't try and start generating hypotheses and you don't have a question yet. Make sure you have a question and then you generate hypotheses as to what the potential answer could be. So you generate an alternative hypothesis, and you've got to generate many alternative hypotheses. You design your experiments to be able to shift them to test different hypotheses, and so that you can exclude certain hypotheses. It actually becomes an exclusion exercise. And then you perform the experiment, and then repeat. You recycle the procedure. You continuously go until you really refine it. Okay, so there's just an example of the two major approaches. You'll probably, if you are on a hypothesis-driven, if you are in this kind of sphere, you'll probably only have one hypothesis that you're testing. But if you're in the hypothesis-driven, see if you can use this multiple competing hypothesis framework, because you'll find it's very, very useful for formulating your understanding of your study system. And I also just want to say that when I was looking this up, trying to look at some old research, I came across this Wikipedia article with analysis of competing hypotheses, which is an unbiased methodology for evaluating multiple competing hypotheses called Conserved Air. It was developed by Richard J. Heer for the Central Intelligence Agencies in the 1970s. It's rubbish. They claim that they generated this, but they actually just started right out of science. The reason they did this and they were using this multiple competing hypotheses was because someone would see the observed data and they had their pet hypothesis, they could fit that data. And then that's what they went with it. They were right. They didn't look at any alternatives. So what they try and do now is generate a range of alternative hypotheses in a scenario or under a question and see which ones the data fit and which ones the data can exclude. And we take away the ones that the data can exclude, and then you've got to keep on the table those hypotheses that aren't excluded, that fit with the observed data. And then you've got to go look for other data to work out which is correct. So the CIA had a massive problem with biases. Everyone would have their pet theory, use the observed data, and then that would fit. But this was developed to overcome that. You can see why it's useful in science. Yeah? So if you don't run with your pet theory, you don't run with the rodent delivery hypothesis like I did initially. That's so excited about the system. I have been, it's taken me a long time to think about the system and generate alternative hypotheses. But that's all part of good science. I'm going to cover on this just a little bit, but I also want to highlight this to you. So, replication, the foundation of science, yeah? Absolute foundation of science. I would argue that that's what differs science from any other field, or any other mode of thinking, is this idea behind replication. But have you noticed in your literature, you guys have been exposed to a lot of literature, and you've been doing write-ups and projects and whatnot. Have you noticed that there's always replication within studies? How many times have you come across replication among studies? And what I mean by that is like, there was this study performed, and then someone else performed the exact same study, and then someone else performed the exact same study, so that you have replication across studies. Once or twice. It's rare though. Yeah?
Another thing that's rare is long-term studies.
Yeah, long-term studies are incredibly rare. Generally because most studies are linked with a PhD. Because PhD funding you can get funding for. PhDs you can get funding for. But our funding cycles run 3 to 4 years. And there's no guarantee of getting funding for a 20-year project. There aren't a 20-year funding project, so long-term monitoring and long-term studies are very rare because they also cost a lot. The point that I want to make with this is that if you find a nice study that fits your topic quite well, don't feel abashed at replicating it. Or replicating it in a different system. Doing what someone else did in a new system or a new way, and if you get the same result, that's fantastic. You've confirmed something that we think we know in that community. If you get a different result, you go like, well, that paper there, we think we know, I did the same thing, and I got a different result.
You did wrong.
That's a bit of a problem. Try not to do it wrong.
Maybe they did it wrong.
Or maybe they did it wrong. Yeah, you're going to be pretty convinced that you didn't do it wrong. You're going to kind of rule that error out first. So, and again, so you don't even worry about novel research at this point. Don't throw that word out. You want to look for valuable and meaningful research. And replicating studies is valuable and meaningful. So if you find something like, hey, I can do this. We have the equipment, we have whatever it requires to take, I have the time to do it. You can repeat the study. And then, this replication of studies, whether it's good or bad, publishable, it has to be publishable. Repeating something that is adding value to say we get the same result as value, so it's certainly publishable. I'm not going to go through this now, I'll cover this another time. Okay. So we've discussed what you can do to get to know your community, and I'm now going to give you some homework that you build around your project to get to know your community that your project lies within. So, I want you to identify some key researchers. 5 key researchers that have written on your topic, who are leaders in the field. I'm going to email this all to you guys. Just this slide. I will email it. Just this slide. And I want you to write up a short bio on them and how they, and I'll copy and paste from their website. Usually pretty obvious. These insults our intelligence. I want you to go like, this guy studies these things that are related to this project. Yeah, you need to relate this up. So, some scientists talking. You're welcome to email them on their views on their project. And then I want you to dive into the literature. I want you to do that skimming exercise. I want you to look at speed read, skim through quite a lot of literature to find at least 20 that are relevant. Wait. Yes. Now, because you want to know stuff that's relevant now, because you want to design your project, or you want to co-design your project with the supervisor, knowing what it is that your community thinks and what is important for them to know. And I don't want you to, if you find a really useful paper, I don't want you to go on and on and on and talking about how amazing this paper is. I just want like 100 words to say like, this is a review of this topic that covers the full spectrum of all the work that's been done up until 2008. It's missing this component, which is in my project. Or, yeah. Something along those lines. It doesn't necessarily have to be it's missing something, but it could be saying my project fits into this review is an understudied part of this review. Because all the other papers will be something else, or yours could be in a well-studied part. So 50 to 100 words in how you interpret that paper in your study. Please, this also is a referencing exercise. The one massive complaint that I got back from all the supervisors who were reading through the short essays was that the referencing wasn't correct. People were just copying and pasting the referencing, and some of the lecturers felt like crying because you should know this, you should know about referencing. We shouldn't even have to check it. You should just know it. So there are actually going to be marks for the reference. And, easy marks. Just get the referencing style right. And we don't care what referencing style it is, as long as you're consistent. You can choose whatever referencing style you want. Be consistent. That's the only thing you've got to do. Except for the nature one, which is like somebody et al. In the year and maybe an abbreviation of the journal, which is absolutely useless because we don't know what the title is. We need title, journal, issue, etc. Okay. Page number. And then, just so that I don't forget about that.